Okay, so this is chapter 12, and we are talking about neural tissue. So the first thing we're going to do is talk about how the nervous system is organized, and then we're going to jump right into the transmembrane potential, right? Which is how do we generate an action potential, which is a nerve impulse, and then how does that get transmitted to another cell, okay? So first of all, the nervous system, it's kind of like it's electrical wiring. I know that when I asked for the student information, a lot of students said that this is the most difficult. I have to say that this is probably the test that the students did the best on. So I think that after we explain what's going on, it's going to be much easier for you to understand, all right? So, you have your brain, and the brain is like the control center. It's going to be doing all of the information processing. It's going to be bringing in all sorts of sensory information is going to be coming into the brain, and then the brain has to decide, what am I going to do with that? Do I need to send it to other areas of the brain, or do I need to just send out a command to these effectors in the body? Okay, so with this information processing, if we look at where it all starts, okay, we look at where it starts, it's all going to start at the sensory receptors, because so far your body likes to be in homeostasis. So it likes homeostasis, means homeo is home, stasis is stable, a stable home. It likes that. It likes all of these things to be in the correct range, calcium in the correct range, sodium in the correct range, body fluids in the correct amount. All of these things should be in a range. And if it gets out of range, then the body gets out of homeostasis, right? So when something goes, when the body detects a change, and it could be that something is going out of that range, it could be that there's a touch sensation, could be, pain. When any type of stimulus is, when there's a stimulus, when there's a change, these receptors, these sensory receptors, are going to detect it. So that's the first thing that has to happen. We have to have sensory receptors detected. So there's three boxes there. So you have three different types of sensory receptors in your body, okay? I'm not going to write them in here. I'm going to let you guys write them in. We have the special sensory receptors. So what are your special senses? Seeing, hearing, tasting, smelling, balance, right? Those things are all special senses. Why do we call them special? Why are those special sensory receptors? Because the cells that detect sight are found just in your eye. They're not in your arm or your leg or all over your body. They're just in your eye. Your hearing receptors are in your ear, right? So they're special because they're in very localized places. We also have then visceral sensory receptors. So what's viscera? Organs. Your organs, right? Your organs are viscera. So they have their own sensory receptors. So your stomach is going to feel pain very differently than your skin would, right? Okay, so visceral is your organs. And then the last one is a somatic sensory receptor. So somatic is going to monitor your skin, your skeletal muscles, and your joints. Right? Skin, skeletal muscles, and joints. So those receptors are detecting any type of touch, tickle, vibration, pain. So we have all these sensory receptors, and they've detected something. There's something going on. The visceral receptors, they might detect a chemical. Maybe there's a chemical change. You just ate something, and now it detects that the pH dropped, or the pH went up because you ate a food that's more alkaline. So your pH goes up. So they detect something, right? And so those receptors are going to stimulate a sensory neuron. So these lines right here, these are sensory neurons. And they're carrying that sensory information. The division that we call this, as that sensory information moves up to your brain, we call it the afferent information. Afferent division. It's the afferent division. A for arriving. It's going to arrive at the central nervous system. It's going to arrive at your brain. Okay, so the sensory information travels through sensory neurons to get to the brain. The brain decides, okay, this is what's going on. This is what I'm going to do about it. And now the brain is going to put out a command, right? So it coordinates a motor command, and that motor command is going to go down through the efferent division. Efferent. So only motor commands go down through the efferent division. That's a command going out to something in your body, right? What we call an effector. And this is where we get a little bit of a division, because the efferent division actually has two different subdivisions. One is called the somatic nervous system, and the other one is called the autonomic nervous system. The difference between those two is that the somatic nervous system is sending a command to the skeletal muscles. Okay? And the autonomic nervous system over here is sending information to all the other effectors in your body, which happen to be smooth muscle, cardiac muscle, glands, and adipose tissue. Smooth muscle, cardiac muscle, glands, and adipose tissue. Okay? Somatic nervous system goes to the skeletal muscles, autonomic nervous system goes to everything else, right? Now, in the autonomic nervous system, there's two subdivisions here under the autonomic nervous system. The one division over here is the rest and digest division, and then we have the fight or flight division. They're both going to send messages to the effectors, but they have opposite effects. What's the name of the rest and digest? Rest and division. It's supposed to say rest and digest, so you can change that. It should say rest and digest. What's the name of the one that's rest and digest? Parasympathetic, right. So the parasympathetic is going to send messages to those effectors if it's been told to do that by the brain, right? The other one, the fight or flight, that's the sympathetic division. Sympathetic division. That is your fight or flight. If you're in a crisis mode, that's the one that's going to get activated, and it's going to send a very different message to those effectors. Okay, now the control centers are going to be different. So the control center in the brain is going to be different for the somatic nervous system versus the autonomic nervous system, right? So the brain, for the somatic nervous system, those commands are going to be coming from an area that we're going to see later is in the primary motor cortex. So the primary motor cortex is the control center that sends the message to the somatic nervous system. And the hypothalamus is the control center for the autonomic nervous system. Right? So the commands are coming from different areas in that brain, in the brain. Okay, so this is a neuron, And we have different parts of the neuron, and I'm going to kind of let you guys do the drawing just because I'm not so used to this technology. And then when I go back and I have my lecture, I'll go in and I'll make arrows and put in words and stuff. But right now I don't want to do that. So you guys are just going to write your own things, right? So let me show you what the neuron is. This is the cell body of the neuron. So in the cell body of the neuron, there is a nucleus. What does the nucleus do? It has DNA in it, and what is the DNA's purpose? To produce proteins, right? It produces proteins. So some of the things that the nucleus is going to produce are neurotransmitters, right? So they're going to produce neurotransmitters. The neurotransmitters are going to get transported down this long thing called the axon, and then they're going to be in these little vesicles down here. And that's what we call, those are the axon terminals, right? Those are the axon terminals. So we've got a cell body with a nucleus producing neurotransmitters that are put in vesicles and transported down, and now all those vesicles are in the axon terminals, right? Okay. We also have, if we look at the one thing that we don't have in the neuron, we don't have centrioles. Centrioles were an organelle. What do centrioles do? They are involved in mitosis. So they help to produce that, spindle apparatus that helps to divide the neuron, right? Or not, I'm sorry, divide the cell. The neuron doesn't have those. So what can't the neuron do? Can't replicate it, can't divide, right? So you have what you have as far as neurons go, right? That's it. Now we look at some other things, and these extensions coming off of the cell body, those are called dendrites, right? Dendrites. What was the purpose of dendrites? Do you remember? So dendrites detect a stimulus. They detect stimuli, right? That's what they do. So they can detect different types of stimuli. There are different types of receptors on those dendrites. And there's also some right on the cell body itself, but most of them are on the dendrites. So there are 3 different types of receptors that are found on those dendrites. We have mechanical receptors, and they're going to detect if one of those dendrites is just bent, like in your skin. They can detect when the dendrites are bent. Or when they're touched in some way, right? If they're cut. So that's mechanical. Okay. We also have the most numerous of the receptors that we have are chemical. And those are the ones that we're really going to be talking about a lot, are the chemical receptors, because they're the ones that that's the one that neurotransmitters are going to bind to. So we're going to talk a lot about those. But there's different types of receptors on there, right? So that's our neuron, and that's kind of all I wanted to say. One other thing, I guess. When those neurotransmitters are traveling down the axon this way, we call that an antegrade movement. They're just moving down so that they can be stored in the axon terminals. But there's also another thing called retrograde movement, and so that is like rabies. If a neuron gets infected, the rabies virus will actually move in a retrograde movement to the nucleus to do damage to the cell body in the nucleus. So that's retrograde. Okay. Now, the transmembrane potential, because that's where we're starting, right? The transmembrane potential is the fact, what it means is that there is a difference in the charge on the inside of the neuron on the cell body versus the outside. That's what a transmembrane potential is. There's a difference. The charge inside this cell body here of this neuron is different than the charge outside here, okay? So we know this because we have equipment that will actually check for the millivolts. So we know exactly what is going on there. So we're going to spend a bit of time now talking about the 3 different types of potentials, transmembrane potentials. We have the resting membrane potential. This is a potential where the neuron is resting and nothing has disturbed it. It's just resting and there's no disturbance. And we know in that cell that the resting membrane potential is a negative, try this, negative 70 millivolts, right? That's what it is inside the cell. So there must be things, there must be things inside the cell that are making it more negative, and there must be more positive charges outside the cell in contrast, right? So what we find on the inside of the cell, on the neuron, is that in the resting membrane potential of negative 70 millivolts, right? First of all, we have a lot of negatively charged proteins. Okay? Now they kind of cluster themselves near the center of that cell body. Okay, but they're making that cell body more negative. Right? Now, we also know that with every cell in the body, we have 2 ions that are going to be very important in creating this transmembrane potential. Do you know what those are? Sodium and potassium, right? So where is sodium higher in concentration? Inside the cell or outside the cell? That's right. So we have a high concentration of sodium outside the cell. Okay? So I put sodium, when I put those in brackets, that means concentration, right? And then there is sodium on the inside of the cell, but it's just smaller. So I'm going to make it a little bit smaller. There's quite a huge gradient with sodium. There's a lot more sodium outside the cell than there is inside the cell. Sodium, in the process of diffusion, when we talk about diffusion, what do we say? Which direction would sodium want to diffuse? Yeah, inside. It wants to get inside that cell very badly because it wants to go from high concentration to low concentration, right? So there are these sodium channels that are always open, and we call those leak channels. And so those leak channels are allowing sodium to move into the cell. It's going to go down its concentration gradient. So we always have some positive charges moving into the cell. If we have positive charges moving into the cell, what happens to the membrane potential, the resting membrane potential? Right, it would get more positive. If positive charges move in, the cell would be more positive, right? But we also have potassium. And where is potassium highest in concentration? Right, so potassium is highest inside the cell. Try to lower this so I can write better on it. All right, so we have a high concentration of potassium inside the cell, and we have a little concentration of potassium outside the cell. So based on our rule of diffusion, which way does potassium want to move? Out. So if potassium is carrying its positive charge out of the cell, what's going to happen to the inside of the cell? It's going to become more negative. Positive charges, potassium's moving out, it's taking its positive charges with it, and it's making the cell more negative, right? So you think, well, okay, you've got positive charges moving out and positive charges moving in, but we said that our resting membrane potential is a negative 70. The reason for that is that there are more potassium channels, leak channels, than there are sodium leak channels. which means we have more potassium moving out than we have sodium moving in. Yep.
Why did you say there's more leak channels?
It just is. There are a set number of leak channels. So we could say that the cell membrane is more permeable to potassium than it is to sodium. It's just the design of the cell membrane. Yep. So we have more potassium moving out, more potassium moving out than sodium moving in, right? So it's those factors then that create that resting membrane potential. It creates that potential of a negative 70 millivolts, right? Now, this happens all the time. It's always going on. The leak channels are always open. So if they're always open, what's going to happen to the inside of the cell? It's going to fill up with sodium, wouldn't you think? Sodium's leaking in all the time, and it's losing its potassium, right? Potassium's leaking out all the time. They are always open, those leak channels. So we have to have something that will correct that, right? We need something that will correct it. And we have what's called the sodium-potassium exchange pump. So there is a big old protein scattered throughout that neuron and throughout those other cells in your body. And it's a pump. And it requires ATP. And it will pump out 3 sodium, and it will pump in 2 potassium. So it's trying to bring the potassium back in, and it's trying to take the sodium back out so that we can stay at that resting membrane potential. Otherwise, you got a cell filled with sodium, right, and lacking potassium. Okay. Everybody good now? Everybody so far? Okay. Yes.
You said 3 potassiums going in and that.
2 potassiums in, 3 sodium out. I wish a student in the past gave me I forgot it. I have no idea what it was, but it was really good. I can't remember. You just have to memorize. It pumps 3 sodium out and 2 potassium in. Okay. Okay. So, one of the things that we know about diffusion, there are several things that can affect the rate of diffusion. Like even though those channels are always open, there's some things that can always speed up or slow down the rate of diffusion, right? So if we had a bigger concentration gradient between the inside and the outside of the cell, if it was bigger, if there was like no potassium outside the cell and you had, still the regular amount of potassium inside the cell, the rate of diffusion would be faster, okay? If the potassium was, the same outside the cell as inside the cell, it would slow down diffusion. So I'm saying this because I'm going to ask you a question. And I'm going to get rid of all these things. And I don't know any other way to do this. I got to figure out how to do this app. Okay. You have a patient, and your patient has hyperkalemia. Do you know what hyperkalemia is? Hyperkalemia is high potassium levels. So where is that potassium level high? Emia means it's in the blood. So if it's in the blood, it's also in the extracellular fluid. So now we've got a patient that has high potassium levels outside the cell, and they have their regular amount of potassium inside the cell. What's that going to do to the rate of diffusion? It's going to slow it down, right? So it's going to slow down the rate of potassium moving out of the cell, right? Do you understand that? Okay, so you have we know that potassium is higher in concentration inside the cell. We know that. We know that potassium is a smaller concentration outside the cell. And because of that concentration difference, potassium wants to move out of the cell. If we take away that concentration difference, and we say that the patient has a high level of potassium outside the cell, and I think I know why you guys are confused, because I first said hypokalemia, now I'm saying opposite, it's hyperkalemia. If the patient has too much potassium outside the cell, that's called hyperkalemia, right? Too much potassium in their blood. What's going to happen to that rate of diffusion? Does potassium want to move out of the cell? No, potassium does not want to move out of the cell. So if potassium's not moving out of the cell, and we were supposed to have a negative 70 millivolts, and potassium's not moving out of the cell, what happens to that negative 70? Is it going to become more positive or more negative? It's going to become more positive. We're going to come back to this again when we look at an action potential. I just want you to see this on why this happens. Potassium doesn't want to move out, so potassium stays in. So that means our cell is going to become more positive. It's going to go, more positive means closer to 0. So maybe it goes to negative 69. Maybe it goes to negative 68. Maybe it goes to negative 65, right? But it's getting closer to 0, right? Okay, we're going to talk about an action potential in a little bit. You guys remember what the threshold was in an action potential, right? Negative 60, right? Negative 60. So it's getting closer to that threshold, right? Okay. So that's a resting membrane potential. So it's going to be really important for you guys to understand the factors that create a resting membrane potential. So those will be like these are the straight answers that you need to know. What are they? They're the leak channels. There's more potassium than there are sodium. There's potassium is higher inside the cell. Sodium's higher outside the cell. Sodium wants to move in. Potassium wants to move out. All of those factors, those are all things that you need to know on the test, right? So those will be questions. Those are straightforward. And then there's going to be what's going to happen to the resting membrane potential if the extracellular fluid has an increased amount of potassium. Right? What's going to happen to it? Well, it's going to become more positive. Okay, so that's kind of like one of the questions. All right, in the opposite, if we have a lower amount of potassium, that's hypokalemia. Now potassium wants to move out even faster because there's a much bigger concentration gradient than before. What's going to happen to the neuron then? What's going to happen to that resting membrane potential? What is it? It's going to become more negative. So now it's going to go from negative 70 to negative 75, negative 80. It's moving away from threshold. Okay. Right? Okay. So good. So what I want you guys to do right now So we talked about the resting membrane potential. We talked about a graded potential. Now we're going to take it one step further, and we're going to talk about an action potential, right? So an action potential starts as a graded potential, right? So the graded potential is just a baby potential, and then the action potential is like the grown-up potential. So what we had when we talked about the graded potential was that the sodium, the sodium channels, the gated channels, they are chemical gated channels. And why are they chemical gated channels? Because what opened them? Acetylcholine, which is a chemical, right? That's why we call it a chemical gated channel. And because it will only let sodium in, it's not going to let anything else in, it's called a chemically gated sodium channel, right? That's what that is. So all of the sodium is pouring into the cell, and we get all these positive charges, and like I said, they're all going down to the axon hillock. And they're accumulating there. So we're getting more and more and more positive right there in that axon hillock. We're getting closer. We're getting more positive. We're moving away from negative 70 towards 0. Okay? So I just drew a bunch of positive charges. So that's our graded potential. But now let's say there was enough acetylcholine. And acetylcholine, by the way, there's all these sodium channels all over the place, right? They're everywhere. So if there's enough acetylcholine binding to these channels, that means there's going to be a lot of sodium moving into the cell. And that's going to make the inside of the cell more positive. The current carries those positive charges down to the axon hillock. And now, to start an action potential, the voltage in the axon hillock is going to get to threshold, which is negative 60 millivolts. Negative 60 millivolts. That's the charge in that axon hillock, right? Because of all those positive sodium inside the cell. Now, the axon hillock is like a funnel. It looks like a funnel. So it's just accumulating those positive charges. But on the initial segment, on the entire axon, but the very first part of the axon is called the initial segment, we have these other types of gates, of gated channels. We have other gated channels. Instead of chemical gated channels, we now have what's called voltage gated channels. So all over the initial segment and all the way down the rest of the axon, we have voltage gated channels. So voltage gated channels are sensitive to voltage changes. That's what they're sensitive to, voltage. We're talking about voltage here. We said that to start an action potential, the charge inside the axon has to go from negative 70 to what? What's threshold? Negative 60. Negative 60 is a voltage. That's going to stimulate sodium voltage-gated channels to open. Okay? So we have, they're different. I'll do them in a different color down here because they're voltage-gated channels. They're not chemical-gated channels. They're not sensitive to chemicals. They're sensitive to voltage, right? So, one of the questions I might ask you in an exam is, where are the chemical-gated channels found? Well, they're not on the axon. They're on the cell body or the dendrites. Where are the voltage-gated channels found? On the axon, right? So I'm going to ask you those questions, okay? All right. So now these gated channels are sodium gated channels. So they are voltage-gated sodium channels. Okay? Because, you can't see that very well, green's not a great color. They are sensitive to voltage, and they will only let sodium in. That's it. That's why we call them voltage-gated sodium channels. Okay? So they are going to open as soon as that hits threshold, which is -60. Now there's a massive amount of those channels on the axon. So not only does sodium come into the cell, but sodium is going to flood the cell. I mean, flood it. Flood it, flood it, flood it. Sodium comes into that cell. We were at -60. We were at -60. Sodium's coming in. The cell's going to rapidly go to -40, -30, -20, -10, 0, +10, +20, +30, right? That's what's going to happen to that axon. In that little tiny segment, because those voltage-gated channels were activated and opened, this membrane potential is going to go all the way up to +30. There's that much sodium moving into the cell, right? Does that make sense to you guys? Voltage-gated channels are stimulated, they open up, sodium floods the cell. So the charge goes all the way up to +30. Okay? Right? At +30, the sodium channels are going to close. Okay? The sodium channels close. So sodium no longer can come into the cell. So if positive charges aren't coming into the cell, that means the charge isn't going to go any higher than +30. Right? At the same time, though, these potassium channels, the voltage-gated potassium channels, those potassium channels are sensitive to +30. And those receptors are also all over that initial segment of the axon. They're all over it, spread through the whole thing, right? Tons of them. So when the membrane potential hits 30, potassium is going to leave the cell, and it's going to take away with it its positive charge. Right? So if potassium's leaving the cell, taking its positive charge, what's going to happen to that initial segment then? What happens to the charge inside that initial segment? It's going to get more negative, right? So now we're going to go from a +30 and we're going to become more negative. What that means is it's going to go from +30 to +20 to +10 to 0, right? To -10, -20, -30, -40, -50, -60. It's going to go all the way down to -70, okay? And so it's down at -70, and if those potassium channels would have all, so let me tell you this. So at -70, then something else happens. Potassium is also sensitive to -70. Okay? So when this initial segment hits -70, the potassium channels start to close. They start to close. But they're not going to close all at once. So this rapid decrease in voltage is going to slow down. Some of the channels are still open, so that negative 70 is going to go all the way down to negative 80 because some potassium is still leaving the cell. Okay, some potassium still leaving the cell. Finally, it hits negative 80 and all the potassium channels are closed. No more potassium is leaving the cell. It's not going to get any lower than negative 80. And what do you think is going to bring it back up to resting? What did we say maintained the resting membrane potential? The sodium-potassium exchange pump and the leak channels together will bring that back up to a negative 70, which is resting. Okay? Let's look at it. Yeah. All right. Now the sodium channels are a little more complicated than the potassium channels, right? So when we look at the difference between the two, the potassium channel has one gate on it. So that gate will either open or close. It opens or it closes. Very easy. At positive 30, it opens, and then at negative 70, it starts to close. Very simple. The sodium gate, the sodium-gated channel, actually has two gates. It has a gate on this side, and it has a gate on this side, right? One of the gates is called an activation gate. That's the gate that will, it's going to open and close and allow sodium to come through, okay? The inactivation gate also has to be open to allow sodium to come in. All right? If that inactivation gate is closed, you are never going to be able to, you cannot get an action potential to occur. There's no stimulus big enough that's going to cause that inactivation gate to open, right? So here, what we see in this example is we're talking about the resting membrane potential. So at rest, these voltage-gated channels should be closed because at rest, all we're really talking about was the leak channels, right? The leak channels in the cell body. We're not talking about these voltage-gated channels really at all in the axon. So at rest, both of those channels are going to be closed. Potassium's closed and sodium's closed. Nothing has really affected it yet, right? But the sodium channel, if we look at it, at rest, the inactivation channel is open and the activation gate is closed, okay? What that tells us is that if there's a stimulus, if you reach threshold, that activation gate can open. So when the inactivation gate is open, that means there's a potential there to get an action potential, right? Inactivation gate is open, the activation gate is closed, but if the voltage gets to negative 70 or negative 60, that activation gate will open. And the reason we have to talk about this is because this is going to help us understand the refractory periods, all right? That's why we're talking about this. So this has nothing to do with the generation of the action potential. When you're talking about the generation of the action potential, I just want you to talk about sodium gates opening up, sodium gates closing, potassium gates opening up, potassium gates closing. I just want you to talk through it, think through it in those general terms. But when we're talking about refractory periods, And a refractory period is when can you have another action potential, okay? We want to talk about the sodium gates, right? So so this is at rest. Now we look at a graded potential. So now what happened here is that we had a chemical, like acetylcholine, bind to that sodium channel, the voltage-gated sodium channel, and it opened the activation gate. Well, now both gates are open. Sodium can now come in. And we saw that sodium comes in, and we depolarize because positive charges are coming in. We depolarize, right? So that's what happens in the graded potential with depolarization, right? At repolarization, that activation channel will close, the potassium channels will open, all right? But let's go to the next, let's look at an action potential then. See what happens here. In an action potential at positive 30, we said two things happen. We said the sodium channel closes and the potassium channel opens. Here we see the inactivation gate closing, and we see the potassium gate opening, right? Sodium can't come in anymore, but potassium can go out. That's what causes repolarization. Repolarization. And we're going to continue to repolarize until we get to negative 70. And at negative 70, the sodium channels are going to regain their normal resting membrane potential status, which means the activation gate is closed, but the inactivation gate is open, right? At negative 70. So if there's a stimulus, if there's a big enough voltage change, that activation gate can open. Okay? All right? The potassium, at negative 70, the potassium channels start to close. Potassium channels start to close, not all at the same time. So we end up getting hyperpolarization because some potassiums, whoops, wrong. Some potassium still leaving the cell. Some can't because the gate's closed. Some can because the gate's open. All right? So let's look at our chart again, right? So those are the three numbers that we're looking at. What did we say our sodium gated channel looked like at negative 70, at rest? Inactivation channels open, activation channels closed, right? Okay, right? That's what we said. And then we said at negative 60, what does it look like when we hit threshold? Inactivation channels open, activation channels open. And sodium rushes into the cell, and we get depolarization. Depolarization, okay? Then we hit positive 30, and we said that the sodium gated channel is going to close, right? What did we say? Which gate closes? The inactivation gate closes, right? So the inactivation gate closes, And there's nothing that's going to open that inactivation gate. Nothing. Okay? And then we said that then because that inactivation gate is closed and sodium can't come in, and we get potassium gates opening up, we end up getting repolarization, right? Potassium leaves the cell, and we get more negative again. And then we hit negative 70, and what happens at negative 70? The sodium channels regain their resting membrane potential status. So what does it look like? So the inactivation channel will open, but the activation channel closes, right? And that's how it's going to stay. You're going to get hyperpolarization coming down and going back up, but that's because of potassium. Nothing's happening then with that sodium, right? So when we talk about refractory period, a refractory period means there's two different types of refractory period. One is absolute, and that is when another action potential cannot occur at that segment of the axon, no matter how big a stimulus is. That's absolute refractory period. You cannot get another action potential to occur. Relative refractory period means if you get a big enough stimulus, you'll get another action potential. So let's look at this and let's say, okay, when in this scenario will we not be able to have another action potential occur on that membrane? Right here, both of the gates are open. We're already depolarizing. Do you think you're going to get another action potential? No, you're already action potentialing, right? You're already depolarizing. So you're not going to be able to do there. Are you going to be able to do it here where the inactivation gate is closed? No. You're not going to be able to do it again until you get back down here to this negative 70, right? So from here to here, that is the absolute refractory period. Right? You can't get another stimulus to occur. You can't get another stimulus to open to cause another action potential. Right? Let's look at from here to hyperpolarizing and back up. Right? Can you get, now we see at negative 70, we see the inactivation channels open and the activation channel is closed. Could you get another action potential at that point? Yeah. Yes. This is just what it looks like at resting membrane potential. So if the cell is sitting at negative 70, yes, you can get another action potential. What if a chemical brought the inside of the cell down to here? So now instead of resting, you're now down here in about negative 80, but your gates still look like that. Could you get another one? An action potential? Sure. It would take more of a stimulus because now instead of 10 millivolts away, you're now 20 millivolts away, right? But sure, you could. Even if it's all the way down here, the gate still looks like that. Could you get another action potential? Sure. But now you're going from, if we're down to negative 90, now you're 30 millivolts away, right? So it's going to cause, you're going to have a much bigger stimulus. Does that make sense? All right, I want you guys to go home and study this. Okay, so when we're looking at this graph then, we have to use some terminology, right? So this is our graded potential right here where we move up to maybe negative 61 and back down again. That's a graded potential. But anytime you are going more positive, we call that depolarization. So depolarization is going more positive. It's going this direction, right? And anytime we are going more negative, that is called repolarization. So repolarization is going this direction, right? repolarization. And in addition to that, whenever you go below the threshold, sorry, not the threshold, whenever you're going below the resting membrane potential, anything below that, instead of calling it repolarization, we call it hyperpolarization. Hyperpolarization, right? So when we look at the action potential then, when we look at the graded potential, we have depolarization and then repolarization, right? It goes more positive and then more negative. Depolarization, repolarization. Yes?
So then the repolarization, would that end at the resting membrane potential?
It does.
Okay.
Repolarization ends at the resting membrane potential. Anything below that is called hyperpolarization. All right? Now, when we hit threshold in an action potential and we get depolarization going up to positive 30, it's very fast. So we call that portion rapid depolarization because it's happening so fast. Right? It's just going very, very fast. And then it hits positive 30, the sodium gated channels close, the potassium gated channels open, and we repolarize. And it happens very fast because there's a lot of those potassium gated channels. So we call that, well, you can call it rapid repolarization. Most of the time we just call it repolarization. Okay, most of the time it's just called repolarization. Okay. What else do I want to tell you here before I move on and complicate this? Do you want to explain to each other how this happened?